Okay, this is the second video installment going over the presentation that you sent over to me. And what I did was I carried out the numbering up top. This was the original slide number in the presentation sent over to me. And as I started going through this, I started breaking out information. I started embedding information from other sources right into these slides so you can kind of cross-reference them. And I have to say, probably one of the most effective tools that you'd have right now is to use some of the links that I provide, put them on one side of your screen over here, while this gentleman is talking about epithelial cells, and have your notes from your instructor over here, and maybe on the table jot down notes between the two. So what's nice is for these presentations, it's audible as well as closed captioning on here. So let's turn this guy off here. So with that said, I'm going to go ahead and just kind of rip through these next ones for you. And if you look at this, what I did was you have the four types of glial cells, glial meaning glue. So when you look at this, you have these epithelial cells are glial type cells, meaning they're cells that are used to help hold things together like glue. And while it's not an exact representation of what the cells do, because they have many different functions, obviously, but that's how they're classified. And when you are looking at like the epithelial cells, okay, great. They kind of sit there with their tentacles out and they monitor what's going on with the cerebral spinal fluid and kind of get a lay of the land on how things are going in the actual spinal column itself and in the brain itself. And when you look at this, it's literally the barrier between the cerebral spinal fluid and the interstitial fluid. And the other job that it has is it's to also secrete cerebral spinal fluid. Uh, this link out, if you do the controlled click, it should take you right out to a link on the video. The other thing I did was I color coded each one of these and did a color codification circle so it's easier for you to see. I have to presume your professor is going to have some kind of matching question. I know I would put this picture up without any cues and say, A, B, C, D. Please tell me what A is. Please tell me what B is. Please tell me what C and D is, and be able to pick those out from this drawing. And that's exactly what I would do. I'm going to pause this for 1 second. Okay, here we go. Much better. I just wanted to blow this up a little bit so you can see it easier. So I know it's exactly what I would do. I would have this on an exam. I would actually create this as a PDF and put it on a Blackboard exam and have my students match what is what in this drawing. We take this a step further. We have the microglia, small glia glue, so it's small little molecules of glue. And microglia is specific. There's only one type of cell that's referred to as micro. The rest of them are referred to as macro. And if you look at what these guys do, microglia kind of hang out. They hang out and they wait with their tentacles out and hit the video, and I urge you to link out to this. Does a really good job of drawing. I tried to replicate it and I just couldn't do it. I couldn't do it any better than the video. So they do a good job of describing how these little guys kind of sit there at the ready doing this, kind of getting a feel for what's going on. They're waiting for inflammation. And then when they sense inflammation, they go from a resting state to an active state. And they go from this to a macrophage. And you'll hear that pronounced many times as macrophage and macrophage. And it's you have phagocytosis or phagocytosis. You'll sometimes hear, depending upon the dialect of your instructor or how they teach it, you'll hear both of those. But realize what that is to mean is you have this cell that goes from a resting state that's doing this, kind of like getting a feel for what's going on, and it senses there's some type of waste product, debris, some type of foreign object, if you will, in the bloodstream. And it goes into this mode where it transitions from this to where it becomes like this blob that moves, and it finds these cells and it basically takes them gobbles them up, and then once it gobbles them up, it releases them for other macrophages or macrophages to come in and actually pick those up and take the debris off. So they're kind of like on high alert garbage men. You know, they're kind of sitting there waiting for something to come in, and once they see something is there, they immediately switch gears and they go into phagocytosis to be able to gobble these things up and then be able to get rid of them. Astrocytes, now you had brought this up in our conference call, and astro meaning star, cyte meaning cell. It's that kind of barrier in between the blood and the brain, and I have this over here so you can see it. It's these guys right here. And what they do is they kind of line up, and as a membrane, you know, they allow blood to permeate, and depending upon the size of the cell, it will or will not allow permeation. Now, like everything else, you know, these cells actually do kind of like double duty, and you know, they might have, like in this example, structural support. They regulate ions, nutrient, and gas concentrations in the central nervous system. They also clean out the synapses, the cleft that's in between where a neurotransmitter actually has an impulse. It goes across a synaptic cleft, and there's waste byproduct from that. And so they end up cleaning it out. But also, in a way, they form scar tissue, not like keloiding like you'd see with a scar on your from red blood cells when you cut your forearm. But they do create a type of scar tissue that is well documented in the video provided via this. And this kind of gets more into my world in terms of oligodendrocytes and the OGD, jokingly I call it. They produce myelin, and myelin is a fatty kind of lipid material that is used to wrap around a axon. And if you hold on 1 second here, I can probably reach back and get it. And so you have an axon, and the axon itself is what is carrying the impulse, the electrical impulse from one side to the other. And if you look at this, you have a fatty substance that actually wraps around your axon. Got another one here. We have more fatty stuff. There we go. And so what happens is you have this myelination, this fatty substance that's around the axon. Well, it's got to come from somewhere, right? Well, oligodendrocytes, the original OGD, they create myelin. And so an OGD, an oligodendrocyte, can create one myelination. It can't create two myelinations on a particular axon. And this would be a quiz question I would have. But this guy right here can actually do one above him, and if possible, one below him. So you could actually have one of your OGDs, I can squish that in there, well, create myelin on an axon in proximity above or below. Now, an OGD, oligodendrocyte, again, like I said, cannot create two on one axon. That's the only hiccup with this. And I know I would ask that on a quiz question in a second. And with that said, this will then take us into the next concept of what are Schwann cells. And, you know, Schwann cells themselves are in the peripheral nervous system, and you have the peripheral axons. So the axons that are in the peripheral part of the central nervous system, because central nervous system then branches off into the peripheral. And the Schwann cells themselves are what actually cover the peripheral axons. So if you think back to this example of the axon, Schwann cells are what actually cover peripheral cells. And Again, I would probably put this on a quiz question somewhere and look at, okay, you have oligodendrocytes. That's a central nervous system glial cell. Okay, so it's a glue type cell in the central nervous system. Okay, I got it. Cool. Then you go here and you're like, well, wait a minute. We have something else that produces myelin as well, and it's a Schwann cell. So when you look at this and you look at how this works, just kind of equate Schwann cells with the peripheral nervous system and oligodendrocytes with the central nervous system. And with this, it's the same idea. You're trying to have Schwann cells that create this fatty kind of myelinated sheath that wraps around each axon, and it helps insulate it and helps with the conductivity of electricity down the central nervous system. And the way this looks is starts off as kind of a clamshell look here like I have. You can see it represented over here in this rendering, but it ends up, let me get myself out of the way, you end up having the Schwann cells themselves, as they create the myelin, it ends up wrapping around and around and around and around to insulate this axon better and better and better. So they do a couple of things here, but the big thing you have to remember is, and I would ask this in a quiz in a second, Schwann cells are part of the central nervous system. Well, that's true or false? That's false. I would cite that and reference that back to here and say, well, okay, so oligodendrocytes are part of the peripheral nervous system. Well, that's false. Oligodendrocytes, as you see here in slide 20 of your original presentation, are used for producing myelin in the central nervous system, whereas Schwann cells are used for the same process in the peripheral nervous system. All right, so as you get further into this, and this is where it gets kind of interesting into my realm, when you look at the myelination, that's around the axon, it's not like it's one thick thing of insulation all the way across. You actually have little spaces in there. And those gaps in there are called nodes, and they're called nodes of Ranvier. Nodes of Ranvier, nodes of Ranvier. But what happens is the nodes allow for the electrical impulse to, instead of shooting straight down, it actually skips from each node to node to node to node to node. So if it was like this, it would go right down the entire pipe. With the nodes in there, bloop, that electrical impulse actually goes zip, 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 right down the line. So it is crucial when we start talking about myelinated axons that you have in the peripheral nervous system. Those Schwann cells are part of a process. And in training, when I work with athletes, we train athletes to not only get stronger, but we want them to be more explosive. But for them to be more explosive, there is a central nervous system as well as peripheral nervous system adaptation that has to happen in their training. And part of that training is this, getting them to be able to fire their central nervous system more effectively and more efficiently in a better and more coordinated manner, and be able to recruit more muscle fiber quickly or faster by training them. And some of the research will support that you can actually train and have an adaptation to the Schwann cells and the nodes of Ranvier. The big thing you're after here, though, is getting people to fire faster, and that's really what you have here. With unmyelinated axons, if you look at this, you have one big cell that go ahead and wraps around the whole thing, right? And so there's no nodes in between. It'd be one straight piece all the way through. Let me turn this so it doesn't confuse anybody. And it's just one straight piece of myelination all the way down. Okay, so no nodes. No nodes at all. Okay? This would be unmyelinated, myelinated. No nodes, nodes. And with that, I'm actually running out of time. I'm at about 14:40, so I'm going to break off from this presentation. That takes us all the way through slide number 23 in your presentation.